The kidneys are located in the back of the abdomen and comprise three sections: the cortex, the medulla, and the pelvis. Its function is to clean all the blood in the body by disposing of waste products like urea and regulating the concentration of other products like water, minerals, and salts. The kidneys contain tiny structures called nephrons. These are the structures that carry out the cleaning of the blood. Firstly, blood from the body moves into the renal artery. The renal artery branches out into afferent arterioles, which connect to the glomerulus, a structure containing lots of capillaries. Blood is flowing at very high pressure here, since the afferent arteriole is much larger than the efferent arteriole, which carries blood out of the nephron. This forces smaller substances like water, salts, minerals, and urea into the Bowman's capsule and the main chunk of the nephron, while keeping larger substances like proteins and red blood cells in the glomerulus to leave through the efferent arteriole. This process is called ultrafiltration. There are nearly 1 million nephrons in a kidney, and they can be exposed using hydrogen peroxide. In this picture, all of the white lines you can see are individual nephrons. After ultrafiltration, water, mineral ions, glucose, urea, and other small substances are left in the nephron. Some of these need to get reabsorbed into the blood, like glucose. Some need to be partially reabsorbed, like water and mineral ions. Others, like urea, need to be completely disposed of. The different parts of the nephron reabsorb different substances into the blood. After the Bowman's capsule comes the proximal convoluted tubule, which reabsorbs glucose and amino acids. Glucose is necessary for cellular respiration, so all of it is absorbed back into the blood via active transport. Amino acids are necessary to form proteins, so they are also fully absorbed via active transport. This process is known as selective reabsorption. After this is the loop of Henle and distal convoluted tubule, which deal with salt and water regulation. The descending section of the loop of Henle is permeable to water. The extent of its permeability is managed by the hormone ADH, but is impermeable to salts. Water is absorbed by osmosis in this section. The ascending section is the opposite. It is permeable to salts, but impermeable to water. The distal convoluted tubule is also impermeable to water, so reabsorbs only salts. Both the ascending sequence of the loop of Henle and the distal convoluted tubule absorb salts by diffusion. Finally, the collecting duct collects the remaining water, salts, minerals, and urea and transports it to the renal pelvis, where it will be transported to the bladder for excretion. In a kidney dissection, the main parts that need to be identified are the three blood vessels: the ureter, renal artery, and renal vein, the three different layers, the cortex, the medulla, and the pelvis, and the nephrons in the cortex and medulla. Laying out the kidney like this, the ureter can be seen, and once cut off, the renal artery can also be seen. To see the rest of the kidney, cut into it as shown. From here, the medulla, cortex, and pelvis can all be seen. Each kidney contains approximately 1 million nephrons that can be seen by adding hydrogen peroxide to the kidney. Each of the white lines that it reveals are individual nephrons.